Hey everyone, my name is Katie and welcome to the channel where we will do question-based revision to turn exam stress into success. We will be breaking down real exam questions step by step and revise basic concepts. If you are preparing for certification exam, college university exam, course exam, or brushing up your memory, this is the best strategy to make them stick. So grab your notebook and let's turn those questions into easy wins. In this part, we will cover general immunology and cells and organs of the immune system. Ready? Let's get started. Here is the first question. The adaptive immune system exhibits several unique features compared to innate immunity. So we have adaptive immunity, we have innate immunity. So which of the following best exemplifies a hallmark characteristic of adaptive immunity? It's asking for a feature of the adaptive immunity. Immediate activation upon encountering a pathogen. So this is a feature of innate immunity because it will react immediately since it have a pre-produced receptor and pre-produced cells which are ready to combat a foreign microorganism or an incoming pathogen. So this is not a feature of the adaptive immunity. Broad recognition of conserved molecular patterns across pathogens. So conserved molecular patterns are recognized by innate immunity. So these conserved molecular patterns are PAMPs, pathogen-associated molecular pattern, which are recognized by PRR, pattern recognition receptors. So this is a feature of the innate immunity. Innate immunity have innate receptors, which are PRRs. Adaptive immunity have two major types of receptors, B-cell receptors and T-cell receptors. So these pathogen-associated molecular patterns are limited and conserved on foreign microorganisms. So the feature of the innate immunity is recognizing these conserved molecular patterns rather than specific differences or minor molecular differences between antigens. So that makes it non-specific. So this is not a feature of the adaptive immunity. Amplification of response with increased exposure to the same antigen. So amplification of response when there is a repeated exposure to the same antigen is a feature of the adaptive immunity. So in case of innate immunity, there is no augmentation, even though there is repeated exposure to the antigen. But in case of adaptive immunity, there is augmentation or increment of the response when there is a second and third exposure to the same antigen. So this is a feature of the adaptive immunity. And the last one is uniform response to all antigens regardless of prior exposure. So this means uniform response to all antigens is generic response, which is a feature of the innate immunity. So innate immunity have a generic response to all types of antigens. But in case of adaptive immunity, it has specialization. For different set of antigens, there are different set of response. So this is not a feature of the adaptive immunity. Rather, it's a feature of innate immunity. Let's see. So C is the right answer. So this question is about difference between innate immunity and adaptive immunity. Innate immunity is inborn, it's consistent within a species, it's recognized common structural patterns, it have limited set of receptors, it have a generic response, there is no lag time in responding to a foreign antigen, and the main cells which participate in innate immunity are neutrophils, macrophages, dendritic cells, eosinophils, and NK cells. When we come to the adaptive immunity, it adapts after exposure. That's why it's adaptive. There is no ready-made cell. When we encounter the antigen, those cells which express specific receptors, whether it's B cell or T cell, will increase in number. So it distinguishes fine differences between antigens that makes it specific, and features or cardinal features of the adaptive immunity can be summarized as a 3SDM. So the 3S is S for specific, S for self-limitation, S for specialization. D is for diversity. There is diverse set of receptors. Basically 10 to the power of 9 up to 12 set of receptors can be found for adaptive immunity compared to the innate immunity. This is huge. And M is for memory. So adaptive immunity have a memory, whereas innate immunity have no memory. And the major cells of the adaptive immunity are T cells and B cells. And in both cases, we could have humoral and cell-mediated immune response. So let's move to the second question. Edward Jenner successfully vaccinated a child for smallpox using a lesion fluid from cowpox infected maid. So we have a cowpox infected maid, and he produced effective immune response for smallpox by introducing lesion fluid from a cowpox infected maid. So the scientific explanation for this success is the lesion fluid contains antibody against smallpox. Well, if there is preformed antibody in the lesion fluid of a cowpox infected maid and transferred to a child and protects the child from smallpox, this is perfect passive immunity. But this is not the case, because passive immunity will not last for a long period of time, and it may protect the child when he is exposed to the cowpox, but it's not going to protect him from smallpox. Okay? Inflammatory mediators in the lesion fluid act as antiviral protein. There are inflammatory mediators like interferons, which will act as antiviral proteins, but they will be there when we have active infection. But in this case, the child was exposed to a cowpox first, then when he is exposed to smallpox, he is protected. But we will have inflammatory mediators when we are exposed to cowpox and smallpox. So this is not going to work. And lesion fluids containing cowpox virus which have shared antigen with smallpox. So the lesion fluid contains cowpox virus which have shared antigen with smallpox. This may work. So the shared antigen between smallpox and cowpox will induce the same set of antibodies or response which will protect against both viruses. And this is the idea of vaccination. By the way, Edward Jenner is the father of immunology, and then the term vaccine comes from a cowpox which infects cows, and he observed that those maids which milk cowpox infected cows or others are protected from smallpox. Cowpox virus have inhibitory protein to smallpox virus. So this is suggesting the antagonism against between cowpox and smallpox, but the child is not exposed for both viruses at the same time, so this is not going to be an explanation. So let's see. So see the correct answer. So the basic thing in here is types of vaccination. We have active vaccination and passive vaccination. In case of active vaccination, it could be natural when we are exposed to the real infection, and it could be artificial when we are exposed to antigen or the whole microorganism, attenuated microorganism, or different types of vaccines. And in that case, the immune system will act against the antigen as it is in a real condition in case of a vaccine, and will produce antibodies and effector cells which will protect the body when it's exposed for the same set of antigen in the future. That is active. In case of passive, we will introduce preformed effector molecules, basically antibodies, into the body to neutralize the pathogen, but they are not going to last for a long period of time, and they will not protect the body when the body is, or the host is exposed to the same antigen in the future. So that is a passive. Passive can be natural or artificial. In case of natural passive, a mother will transfer antibodies to her child. In case of artificial, there are neutralizing antibodies given as injection in case of infections, or when a person is poisoned or exposed to some chemicals. So let's move to the third question. A researcher is studying immune response to a polyomavirus in two groups of mice. So there is a virus in two groups of mice. The first group consists of a nude mice which have genetic defect resulting in absence of a thymus. So there is a nude mice without a thymus that makes them obviously immune deficient. The second group includes normal mice with fully functional immune system. After infection with polyomavirus, tumor developed in nude mice but not in normal mice. What is the most likely explanation for this difference? So we have two groups of mice. One immune deficient, one is normal. Immune deficient because they don't have a thymus. So the question is about the function of a thymus. T lymphocytes are critical in rejecting tumors caused by polyomavirus. Or this could be the answer because thymus is site of maturation for T lymphocytes. Macrophages are required for tumor rejection in polyomavirus infection. They may be required, but they don't have any direct link between thymus with thymus. So it's not going to be the answer. B lymphocytes play no role in rejection of polyomavirus-induced tumors. Well, B lymphocytes mature in the bone marrow, but they will require the help of T cells, but it's long way and indirect for this experiment. Natural killer cells independently reject tumors without assistance from T lymphocytes. Yes, natural killer cells are a good example of innate cells which are effective against viral infected and tumor cells. But this is not linked with thymus again. So the best answer for this will be A. So in here, the main point of this question is what are the organs of the immune system and what organs have what function and what cells have what function. So we have two major organs, primary and secondary organs of the immune system. Primary organs consist of bone marrow and thymus. So bone marrow is a site of origin for all blood cells, red blood cell, platelet, and white blood cells. And thymus is a site of maturation for a T cell. So after originating from the bone marrow, those cells or lymphoid cells which are committed to produce T cells will migrate to a thymus and will continue their maturation process. So thymus is a site of production or maturation for T cells. In the bone marrow, all the myeloid cells, which means red blood cells, platelets, and other types of white blood cells, majorly granulocytes and monocytes, will mature within the bone marrow. And from lymphocytes, B lymphocytes will mature within the bone marrow. So bone marrow and thymus have a conducive environment for the maturation process of these cells. And the bone marrow majorly have hematopoietic stem cell, which is a source for all blood cells. So when we talk about secondary lymphoid organs, we have spleen and lymph node. So spleen and lymph node are site of interaction for naive cells, naive immune cells, and antigens. So in case of spleen, those antigens circulating within the blood will be screened within the spleen, and B cells and T cells will capture those antigens circulating in the blood within a spleen. Whereas if the antigen is circulating within a tissue space, it will be captured within a lymph node. Remember, we only have one spleen, and there are more than 500 lymph nodes in our body. So lymph node is more effective in launching an immune response because they are large in number compared to a spleen. So spleen and lymph node have partitions. For example, in case of spleen, we have the red and white pulp. The red pulp is site of recycling for old and dysfunctional red blood cells, and the white pulp is rich in white blood cells, and its major function is capturing antigen circulating in the blood circulation. In case of lymph node, we have a cortex, paracortex, and medulla. The cortex is rich in B cells, naive B cells, and the paracortex is rich in T cells, and the medulla is site of further multiplication for those cells, those B cells and T cells, which are already activated by an antigen. So once they are activated within the cortex and paracortex, they will migrate to the medulla, and further differentiation will occur within the medulla. Then they will migrate as effector cells to site of antigen origin in different parts of the body. And when we come back to the primary lymphoid organ, in case of thymus, we have cortex and medulla again. In the cortex is filled with immature precursor cells committed to develop into a T cell, and we call them thymocytes. They will be packed within the thymus. Then gradually, when they finish their maturation step, they will migrate to the medulla, and after they become fully matured, they will be called naive T cells, and they will migrate to secondary lymphoid organ. So to see the highlight of cells of the immune system, we have 5 types of white blood cells. Lymphocytes are the major one. We have neutrophil, eosinophil, basophil, and also monocytes, but there is also dendritic cells. So to have some highlight on the markers, all white blood cells will express CD45. So This counting machines are dependent on that. And we have two major types of lymphocytes, T lymphocytes and B lymphocytes. T lymphocytes will express CD3, and we also have two major types of T lymphocytes, CD4 and CD8. And B lymphocyte will be differentiated from T lymphocyte by expressing CD19 and 21. Neutrophils will have a marker of CD15, 11B, 60B, 66B, and monocytes could also have a marker of CD14 and 64. NK cells have CD16 and CD56. Eosinophils also have a CD66B, 49B, so they can be differentiated by this marker. And basophils, which have dark granules, have a CD marker of 123 and 294. And there is also a mast cell which is functionally similar with basophil, but it's located in tissue. So CD117 will be used, 117 will be used to differentiate basophil from mast cells. So these are just highlights of cells and organs of the immune system. Let's move to the next question. So a patient's flow cytometry report shows the following: total white blood cell count 12,500 per micron, lymphocyte 25%, T lymphocyte 60%, B lymphocyte 35%. So we have the total white blood cell count. We have a lymphocyte. Out of the 5 white blood cells in this much is a total lymphocyte, and we have two major types of lymphocytes, B lymphocyte and T lymphocyte. So the question is the absolute count of T lymphocyte in this patient. So to calculate the absolute count, first we have to know the absolute number of lymphocytes. Then from this absolute count of lymphocyte, we will take 60% of that. So 12,500 times 0.25 will give us absolute count of a lymphocyte. Then when we multiply this with 0.6, it will give us absolute count of a T lymphocyte. So when you do the calculation, you'll get this one. So if it's in exam condition, I will calculate this part. So I will take 1/4 of this and again half of this, 0.5 of this. Why 0.5? Because 60% is just 10% above 50%. So I will guess the approximate value if there is no calculator provided in the exam condition, but most of the time we will have a calculator in the exam condition. So in here we also should know the normal value of the blood count. For example, in case of total white blood cell count, 12,500 is a bit higher. Normally we will expect from 4,500 up to 11,000, but if it is in newborn, it will be way higher, up to 30,000. And out of this total white blood cell count, from 50 up to 70% will be neutrophil, 20 up to 40% will be lymphocyte, 2 to 8% will be monocyte, 1 up to 4% will be eosinophil, and basophils most of the time are almost absent, less than 1%. So T lymphocytes can be CD4 or CD8. So the CD4 and the CD4 to CD8 proportion is most of the time 2 up to 1 or 3 up to 1. But in case of some effect, for example, in case of HIV, since the virus targets CD4 cells, it may revert this proportion. So we have to know the proportion of CD4 and CD8, maybe, if it appears on the exam. And normal value of CD4, the absolute value is from 500 up to 1,500, depending on the patient and region or geographic location, race, and other factors. And in case of CD8, from 150 up to 1,000. And normal value for NK cells again is 5 to 10%. NK cells are one of the innate cells, so we should know these things too. So let's move to the fifth question. A researcher is studying the development of T cells in the thymus. So thymus is site of maturation for T cells, and isolates a population of T cells that express both CD4 and CD8 markers. Thus, T cells are in the process of undergoing positive selection. Okay, it's one of the developmental stage, and must recognize MHC molecule in order to survive. Okay, MHC is major histocompatibility molecule, or HLA, or human leukocyte antigen, and they have to recognize this molecule in order to survive. Okay, otherwise, they will not survive. What is the most likely stage of T cell development for this population? So, what is the stage of this T cell? So, double positive thymocytes, they could be double positive. By being positive, we are taking markers of CD4 and CD8 as a signal. So, if they express both CD4 and CD8, they will be double positive. If they express one of it, they will be single positive. If they does not express both of them, we will call them double negative. So, technically speaking, when a thymocyte or a precursor T cell migrates from the bone marrow to a thymus, it's double negative. It doesn't have a CD4 and CD8 marker. Then it will be packed within the thymus. Gradually, they will express both markers, and we will call them double positive. And naive T cells are those who have completed the maturation stage and ready to migrate to secondary lymphoid organ or packed within secondary lymphoid organ. These cells does not complete their maturation step yet. CD4 T cells, well, they have both CD8 and CD4, so they are not single positive. CD4 cells are single positive. Effector T cells. Effector T cells are those activated by specific antigens and targeting that specific antigen to destroy it or to remove it from the body. Those are effector T cells. After activation of a T cell, they will create effector T cells and memory T cells. That is within secondary lymphoid organ, it's not within a thymus. So the appropriate answer for this will be A. So in here, what are the maturation steps or what will happen within the primary lymphoid organ? So those precursor T cells will migrate from bone marrow to a thymus, from bone marrow to a thymus. Then when they reach the thymus, they are double negative. Then gradually they will start expressing CD3 molecule. Then they will express both CD8 and CD4. They will become double positive. Then they will migrate gradually to the marginal region of the cortex and the medulla. Then they will become single positive. So when they are double positive, they will be tested for self-MHC molecule and antigen by thymic epithelial cells, macrophage, and dendritic cell within the thymus. Then if they pass this test, they will pass the positive selection. Positive selection technically means those cells which shows positive reaction will pass the test. So these cells have to recognize the MHC antigen complex. If they recognize the MHC antigen complex, that's positive reaction and they will pass a positive selection. Next to that, they will be tested for strength of interaction. So if they have a strong affinity or strong interaction with MHC antigen complex, they will fail the test. Because if these types of cells migrate to the periphery, they will lead to autoimmunity. So that is a negative selection. In case of negative selection, those cells which shows negative reaction will pass, and those cells which shows positive reaction will fail. So they should not show strong interaction or affinity. They should have weak interaction. So that is about positive and negative selection. So hopefully this session was helpful in preparing you for the exam, and until we meet next time, see you.